All right, thank you.
Okay, good. Thanks, Marta. So what I'm going to do now is, so this is the schedule we have. We have two last things, okay? Bit more on modeling, and then Lucian's going to talk about SPML and biomodels a little bit more. That may be a bit, you may not use all that time. We'll finish whenever we finish, okay, everybody? So, but we got plenty of time. Okay, I'm going to go back to the Google Drive, and I'm going to pick up this one. I think that's the one. It is. Now, by the way, yesterday I converted this entire thing to a PDF with lots more notes, okay? So, and maybe I'll make this bigger as well. So if you have any questions that I haven't answered, I could add it to this document actually. But anyway, for now, let's go to the Python notebook. So first thing I'm going to do, as before, by the way, when you come back to this, say tomorrow or something, you'll discover that it's forgotten all about Tellurium. And so you have to reinstall it again, okay? So Colab has a habit of forgetting things quite easily. So I had to reinstall Tellurium here again, okay? And as Lucian said, the minus Q just means just be quiet about this, okay? I don't want to hear everything you're saying because it will actually print out a lot of stuff.
Don't forget to share your
And
screen.
oh, that would help, wouldn't it? Let me start again then. Okay, so we're gonna start with this using Tellurium. What I meant to say was I've copied all this stuff to this document, modeling with Tellurium, and I've added a bunch of more notes and a contents page. And I'll probably, if there are any questions that people ask that's not answered here, I can augment this document with more information. Okay. So, but but this is the sort of notebook version of that. Okay. So as I said, Copilot tends to forget, and so I had to reinstall it. Okay. I had to do a fresh pip install. And as before, I'm going to have to do a restart. So you have to go to runtime and then hit restart. Now, I should add that if you're using a desktop tool, you don't have to do a restart, okay? This is a Colab peculiar thing, all right? So okay, once I've done that, it should be ready to go. First thing I'm going to show you is just an overview of what the whole thing looks like. So this is Python on the right, and Python has all the usual stuff you might want to use, like NumPy, the plotting library, you know, anything else that you like to use in Python. SciPy is the scientific library. SymPy, if you're interested, is the symbolic library like Mathematica. And there's a bunch of other stuff that you can add to it. I should have added Cobra Pi here. Some of you mentioned topology analysis, and somebody mentioned that's CE Plex solver. That's a linear programming thing. We don't do any of that, but something like Cobra Pi will do that. And a lot of these other systems biology tools will live quite happily with Tellurium. Okay, so you can install all these as well if you want. Now, Tellurium itself is a fairly biggish thing. So it supports standards like SPML. I'm not going to mention these other ones, but SPML is one of the main standards. It supports a bunch of modeling stuff like antimony. And then this thing, libRoadrunner or Roadrunner for short, that's the underlying simulation engine. All right, that's all that is. And then it has a bunch of other stuff. Oh, there's Cobra Pi, which I'm not going to mention either, but there's other stuff it supports too, if that's of interest. Then it has a bunch of utilities like visualization, parameter scans, file utilities. And then Roadrunner itself supports ODE simulation, which we've already seen. It also has a lot of metabolic control analysis built into it, if that's of interest. It'll also support some basic stochastic modeling. And a couple of you mentioned topology, network topology. It has a bunch of topology stuff as well, if that's of interest. But I'm going to primarily just talk about ODE simulation, which is what most people do, okay? All right, so that's the sort of overview of Tellurium. The things that we will look at is today and tomorrow is SPML, antimony, Roadrunner, some utilities, and ODE simulation. Okay, and a couple of basic Python libraries. All right. So let's start from scratch, shall we? So you're only going to have to learn 1, 2, 3, 4, 6 keywords. That's it. Load A, simulate, plot, reset, steady state, and another plot thing called plot array. That's it. 6 things, okay? Now there's a lot of other stuff in Tellurium and Roadrunner, but these are the basics which will take you, a long way, okay? First thing, as Lucy showed, is we have to import our package. So I'll do that. Thank you very much. It's imported. And as before, I'm going to shorten the name to TE because typing Tellurium all the time is a real pain. And for some reason, I always get, when I type, I always swap the R and the I, and I end up with typos. So TE is the one, okay? All right. And I'm just going to go over again, just to reiterate again what Lucian did, just quickly. Here's probably the simplest possible model, okay? And this model we describe in a string. Now you'll notice it's got 3 little quotation marks. So it starts with 3 little quotation marks, ends with 3 quotation marks. The 3 quotation marks is really important. It's a way of typing in a Python string over multiple lines. If I had double quote, if I had the normal string, like just double quote, my entire model has to be on 1 line, which can be a bit of a nuisance. But Python lets you do multi-line strings by having triple quotation marks, okay? That's why I'm doing triple quotes. So I can spread the model across multiple lines, okay? Now, the other thing, this is just a normal string, but it's on multiple lines. I'm giving it to a function called load A. And load A stands for load antimony, okay? And this load A comes from Tellurium. As Lucian said, you're more than welcome to type that, but I wouldn't bother. Just shorten everything to TE, okay? And the other thing to note is I have an equal sign here because this load A returns to me the model. Now, the way the computer scientists call this, they say this is a Roadrunner instance. I'll just call it a model. R is my model. I can call it anything I want. Lucian called it RR, standing for Roadrunner. I could call it my model. I could call it liver model. It doesn't matter. You call it whatever you want, okay? I just call it R because I don't like typing too much. Right? So that's my model, R. Everything I do from now on is on R, okay? And one of the first things you want to do, I'm going to get rid of this. One of the first things you want to do is run a simulation, and you do that with simulate. And this, so I'll do that. Oh, hold on. Again, you got to watch notebooks. You got to make sure all your cells are run. Then I'll do that. And look, it just spewed out a whole bunch of numbers. This is the raw data that's coming from the simulation, okay? And the raw data is an array. And the first column will always be time. And the second column, the remaining columns are all the reaction species that can change in time. In this case, I've only got 2, S1 and S2. And so it lists out S1 and S2, all right? Now, because I don't want to see all that raw data, I'm going to capture the raw data in a variable called M, just because I don't want to see the raw data, okay? All right, so there's that. And then, of course, I want to run a simulation, and I do that by running r.plot. Now, I don't have to do this in multiple cells. I can do this in one cell, like that. It's the same thing, okay? All right. Let me get rid of this then. All right. Now, the first thing you might say is, yeah, hold on a minute. I want to know what happens after 5 time units, because this is only simulating from 0 to 5. Now, the units here depend on the units for your K1, but I'm assuming they're per second, okay? I'm assuming those units are per second. That means this x-axis will be seconds. So this is 5 seconds of simulation time. But of course, you're asking yourself, yes, but I want to know what happens after 5 seconds. Okay, to know what happens after 5 seconds, I'm going to go back up here, right? And simulate takes a number of arguments, one of which is the start time, and another one is the end time. So this says simulate from 0 seconds to 45 seconds. If I do that, I get a much more reasonable plot, okay? All right. The other thing I don't quite understand. Oh, yeah, okay. Yeah, I know why I don't understand it. I'm going to take this and put it with the model. You notice that the orange line, S2, is starting at about 8, and the blue line is starting at about 2. Even though I said that S1 should start at 10 and S2 should start at 0. What's happening here is, as Lucien mentioned this, it's continuing the simulation from where it left off, right? I don't want to do that because that can be potentially confusing. And I'm going to stick the whole thing into the same cell. Now what will happen is you'll see a much more reasonable run. Now it starts S2 at 0 and it starts S1 at 10, okay? Just to show you what I meant by that, it continues where it left off. If I take this without the model and just ask it to do the simulation and the plot again, you'll notice the difference. You'll see that it basically continues from where it left off last time. Okay, and it left off at about 10, so it's starting off at about 10. And here it stopped at about 0, and so it left off at about 0, okay? So this is done on purpose. We do this on purpose because it makes it easy then just to do another simulation to see what happens. All right, without having to redefine the model again. All right, so if you wanted to start at the same place every time, just put the model with the simulate, and then it gets the that initialization every time. Now, on the other hand, you might not want to do that, because this might be a huge model, and you don't want to drag the model everywhere you go. So there's a quick way of making it start up from where it was before, and Lucien mentioned this, is to ask it to do a reset. What that means is it resets all the variables in your model, S1 and S2, back to where they were at the beginning. And they were, at the beginning, they were at 10 and 0. If I run this one, we should get the curve that looks like what it looks like above. So if I do this, you get the same curve, okay? So reset is a very handy little method that'll reset everything back to where it was before. So we've already covered load A, simulate, and reset, and plot, I guess. All right. So the only other thing I'll add to, yeah, so let's just carry on now. So that's where we are at the moment. We've extended the simulation time to 45. I can extend it, as long as I want. All right. It goes all the way to 500 now, if I want. And you might also want to look at the raw data. I mean, this is just a plot. What about the raw data? I want the raw numbers. So to get the raw numbers, I will assign it to a variable. So m equals r.simulate, and I can print m, and here's the raw data. Okay. So you can save that to a file. Those of you who are pandas people can move it to a pandas frame, which I'll show you in a minute. You can do whatever you like with this raw data. It's up to you. In fact, the next one is, those of you who prefer pandas frames, if you don't know what this means, don't worry about it. But this is how you do the conversion, okay? So those who don't know what a pandas is, it's basically like a spreadsheet. It's Python's version of an Excel spreadsheet, okay? To do that, you need to import pandas, and traditionally it's pandas as PD. And then you have to create your data frame just by going PD data frame, the data that's come from simulate, and the columns. So the nice thing about pandas frames is all the columns and rows can be labeled. You give it the column names, okay? And then you can do whatever you like with it as a pandas frame. One, for example, you can save it to a CSV file, right? So as a CSV file, you can load it into something else, like Excel, right? You can load this directly into Excel. You can, if you like MATLAB more, you can load it into MATLAB or whatever. But let me just point out though that these arrays are special in that they have column labels, right? We've done that on purpose, so it's easy to figure out what column is what. The first column is time. Second column is the concentration of S1. Third column is the concentration of S2. All right. Okay. Now you may be saying, yeah, but hold on a minute. There's far too many numbers here. I really don't want all these numbers. I want far fewer. What do I do? Well, there's another number you can add to simulate, which is the number of data points. So if I run this, and here I've only asked for 10 data points, I just get 10, literally 10 data points. All right. So let's say I just wanted 3 data points. I'll get 3 data points, right? And so on. Okay. So you can specify the start time. That usually isn't that important for most models. The end time and the number of data points you want. Okay. Now, if you want, you can put 3 million in here. All right. Don't recommend it, but you could. Okay. So 100 is a nice number. All right. So if I run that, I get 100. All right. Okay. So at this point, now I want to show you. So now that this is the real basics of modeling, right? The real basics of using Tellurium. I've got here a note, show the spider demo. So most of the time, I don't work with notebooks. I don't work in the Colab. People in my group, they use all sorts of different tools. I mean, some people do use Colab. Some people use Spider, like myself. Other people use PyCharm. There are lots of tools you can use to use Python, and I'm going to show you Spider. So this is Spider, okay? And I'm going to clear some of this stuff because I don't want it to. Let me clear this down here. I'm gonna do the same thing, okay, like we just did before. This is what Spider looks like. This is a desktop application. You can download Spider, pip install Tellurium, and you've got this thing, all right? So this thing has, you can see it has a number of panels. It has an editor here. So this is not like a notebook, okay? This is actually more like MATLAB. It's got a notebook here. There's a whole bunch of tabs here with different models I've been messing with, okay? But this is a new one I just made. There's an empty space up here on the right. That's where the plots will appear. And this is an immediate Python console, okay? So I can go 3 + 4 and I'll get 7, okay? When I type new, I've arranged it myself so that it brings up this template for me, all right? And the template for me is it brings in the imports straight away. It gives me a ready load A. And because I know I'm always going to run a simulation, it puts in a simulated plot ready for me, okay? All right, so now I can immediately start typing my model, okay? So let's do the same one we've done before. Okay, I don't know, I need to put in some initial conditions. By the way, if I use a semicolon, I can put lots of things on one line. That's it. Okay. Now if I run it, I get the same as before. Okay. I just like working in environment because I can go back to this now and change it. It's going from 0 to 15. Let's go to 50. Oh, I should have mentioned to run this, I go to the toolbar at the top and click the green button. And again, I get that. What I like about this is no ambiguity about whether I've run any previous cells or not. Also, I can have lots, if you're like one of my kids who has like 1 million tabs open in Google, I can have 1 million models open here as well, okay? although it gets a bit unwieldy after a while. But you can have lots of tabs open which have different models or different variants of your model. You can cut and paste between them and so on. So I just wanted to show you a demo of this model, of this environment, just to see what it looks like, okay? So I'm now going to go back to, are there any questions on that? Otherwise, I'm going to go back to the...
I put the link to our Spider and Tellurium bundle for Windows machines.
All right, good. Yeah.
In this chat.
So that was a quick spider. I just wanted to show you there are alternatives you can use as well. Okay. All right. So I've got this model. Where's the model? Here is the model. There it is. I'd like to get some more information about it. What kinds of things can I get? So these are some new commands which I didn't list in the above list, but I can get some other stuff about it. In fact, let me bring the model back down here. I want to bring the model back down. And I'm going to expand it a little bit as well. Let's do it here. But what I can do, of course, is I can just grab this model from here and bring it into the notepad, right? That's possible, okay? Okay, what kinds of things can I ask about this model? There's a ton of stuff you can ask. If I go to, let me run this, it is run. If I go r dot, lots of things come up. There's lots of stuff you can ask about this model. Let's look at some basic stuff, shall we? If you want to get information, there's lots of get methods. In fact, you saw one to the Lucian. He got antimony and he got SBML, but there's lots of gets. Let me show you a couple. Actually, let me do. Oh, hold on. Let me just bring that up again. Come on. Okay. There's the get. Let me type R. So it reduces me to the get R's. Let me type E. That reduces me to the get E, RE. What I'm going to look at is, let's have a look at the reaction rates. Oops, I'll run this. This gives me all the current reaction rates. In other words, the rate through this reaction and the rate through that reaction. Now, at the moment, because I haven't run a simulation, we're at time zero, effectively. The rates don't look that interesting, okay? It's 1 and 0. And it's 1 because K1 is 0.1, S1 is 10, so 0.1 times 10 is 1, so that's why it's 1. S0, S2, sorry, is at 0, and so is S3 at 0, because I haven't initialized them. And that means the rate through the second one is also 0, okay? Let's do a little simulation here. Oops, sorry. I'll simulate for just 1 second, okay? Let's run that. Now, if I ask for the reaction rates, they're different. I assume you can, should I just zoom in a bit maybe? I should have. They're different. It's 0.904 and 0.246. That's because we're now looking at the model after 1 second. And of course, S1 has changed, S2 has changed, S3 has changed. So the rates are changed, all right? So I can easily get the reaction rates. What happens if I wanted to know, let me put in a new cell here. I wanted to know the current concentration of S1 and S2. So I would just literally print R.S1, R.S2. And that gives me, these are the current concentrations of S1 and S2, right? So when you load a model, the model R automatically gets all the names of the species. In fact, it gets all the names of the parameters and a bunch of other stuff too, okay? So it's really easy to interrogate what's going on in the model at any time you need to, okay? I can also get this method, get floating species concentrations. This lets me get all the concentrations in one list, or array technically. Here they all are, S1, S2, and S3. Now you might be asking, yeah, but hold on a minute. how do you know the first one refers to S1 and the one next one S2 and the third S3? I mean, it looks like they would, given the order of the names in the model, but that you can't be absolutely sure about that. So there's actually another method called floating, get floating species IDs. These are the names, and that's the order of the names, S1, S2, and S3, and that's then the corresponding order here, okay? All right, so if you're not sure what the order is, you can get the order just by asking for the names. Now you may be, okay, so I'll come back to the floating in a minute, okay? I can also get that's the rates of change. I think we did that. Oh no, we haven't done that. If I want to know, well, how fast are these things changing, right? How fast are they changing? I can use something called get rates of change. Let's run that. And these are how fast S1, S2, and S3 are changing, all right? Notice that S1 is changing in a negative direction. That means it's decreasing. The other two are positive. That's because they're increasing, okay? So there are a whole bunch of methods like this that you can use to interrogate the model, okay? All right. Okay, so this model had just 2 steps. Let's make it a little bit longer and let's build a little bit of a bigger model. This time we have 3 steps and there's 2 new things I'm doing here. First thing, I've got this funny thing here, J1 colon. What this is doing is it's letting me name the reaction. So I can name all my reactions. This can be useful for 2 reasons. One, you'd like to give the name to a reaction so you know what it is. But secondly, once you give a name to the reaction, it's possible now to ask it about rates through those specific reactions. So let me just run this for a second. Right, so that's that. And then I'm going to add another code here, and I can actually print r.j1. J1 is the name of the first reaction. If I do that, it actually gives me the current rate through that specific reaction. As you can see, it's very small because everything's gone down to nothing, but that's the current rate of the reaction. And I can name all of them, not just one. I can just name them all. And you can name them whatever you want. I can, I can call this uh I don't know, cat. There you go. If you have any cat lovers out there, I can call the reaction cat, okay? And so I can ask for, if I run this, I can now ask for what's the reaction rate through cat or dog if you're a dog lover, and it gives me the rate through cat, okay? That's the first thing. The second thing is this. You notice the last one, there's no name. I didn't bother to put a name. You're allowed to do that. You can miss out either a reactor or a product. What you're not allowed to do is do that. Okay, that's not allowed. But you can do that. And the reason for doing that is I just really don't care about where S3 goes. I mean, I could put S4 there, right? But I actually don't care. So I'm leaving it out. Sometimes that's convenient to do, all right? So I'm just showing you that it's possible, right? Most of the time you'll put something there, but sometimes you just don't care and you just don't want to put anything, all right? So that's 2 new things we've got there. The name of a reaction and the fact that you can have an empty, so I can actually put an empty one here too. That's allowable too, okay? Now, the only issue here is the S through S1, but S1 is declared down here, and in fact, S1 gets treated as a parameter because it's not there. And if I do that, you'll notice the graph changing. Watch. There's only 2 things now, whereas before there were 3, okay? So let me put that back. And now it's back to there again, all right? One thing you notice here, if you look very carefully at the graph, you can see these straight edges on especially on the orange. Can you see that? That's because I'm not generating enough data points. And so it doesn't look smooth, all right? So what do you notice? Two things. The curves don't look smooth. That's because there's not enough points. So let's fix that by sticking in more points. Let's stick in 200 points. Okay, now it's nice and smooth, okay? This can be really important if you have fast changing dynamics. And you need to capture those changes that are occurring fast, okay? The other thing is everything seems to go to 0. I mean, that's not like a real biological cell. I mean, if a real biological cell did that, it would die, I think. So this doesn't seem like a realistic biological model where everything just goes to 0. And it most definitely isn't a realistic model. So the other problem is to do with the fact that we have a closed system of irreversible steps. There's no interaction of this model with the outside world. It's like the tank model where we don't have a pipe coming in at the front. If we didn't have a pipe coming into the tank model, well, first of all, there's no water coming in. But even if there was water in the tank to begin with, it would just empty out. Not a very interesting tank model. And for that reason, this isn't a realistic biological model. We need to have some kind of input to the model to make it realistic, right? So as a living being ourselves, we have inputs. We're breathing oxygen, and at lunchtime or supper time, we're going to have some food, right? So we are always interacting with the outside world somehow so that we maintain some kind of steady state. Now, how do we do that in a model, though? Well, to do that, we need to have what we call fixed boundaries. Okay. Now, in the tank model, the fixed boundary, if you can think back, was Q1, which is the rate of water coming in to the tank. That was the fixed boundary, okay? Boundaries don't have to be fixed, but we do need to be able to control them. And the easiest kind of control is to literally just fix them, okay? So we will add fixed boundaries. And the quick way to add a fixed boundary is to add a dollar in front of whatever the thing is you're fixing. Now that may not be easy to remember. So there's another way of adding a fixed boundary, and that is just to use the word X for external. And when I go external S1, that's effectively fixing S1. Now, what that means in practice is it just means that S1 doesn't change in time. That's all it means, okay? So if I set it to 10, as I do down here, it will be 10 forever. Okay. All right. So here's the model. I'm using the dollar sign here. I fixed S1. Oops, fixed S1. And I'm going to run it. Whereas before I got this, now I get this. Looks very, very different. Okay. Now it doesn't bother to plot S1 here, as Lucian mentioned, because it doesn't change. It doesn't change. So it doesn't bother to plot it. It's only plotting the things that are changing. And the only things that are changing are S2 and S3. So it's this one and this one. And you notice they change, and then they reach plateau, and that's the steady state. So this really has reached a steady state, okay? Now, there's two special things about the steady state. The first thing is that these rates, the rate of change of S1 and S2 is 0. This is a plateau. It's reached some kind of constant. In fact, let's go from here. Let me run this, then let's have a look at some of these numbers. So remember before I was able to get the rates of change. There we go, get rates of change. We expect the rates of change to be 0. And they pretty much are. I mean, it's 10 to the minus 8 and 10 to the minus 7. So the rates of change of S1 and S2 have really got low, and they're pretty small. I can make them smaller if I ask it to run for a lot longer. So it's now running for a lot longer. And now if I ask for the rates of change, now it should be even smaller. 10 to the minus 9, 10 to the minus 9 is a bit smaller, okay? But effectively, they've gone to 0, all right, the rates of change. The other thing that's very characteristic of the steady state are the reaction rates. I'm going to name them here. Let's print out J1, J2, and J3. What did I do wrong? Oh yeah, sorry. Yeah, this is a notebook issue. I have to make sure I rerun the cell and then run that. Okay. Okay, look at these numbers. 1, pretty much 1, and another 1. The rates are not 0. So what this is saying is through this pathway, S1 to S2 to S3, there's a constant rate of mass flowing through this pathway. That's another characteristic of steady state. So the two characteristics are all the rates of change are 0, nothing is changing anymore, but the reaction rates are not 0. Okay, so those are the two key characteristics. Okay, so rather than me going on and on, I'm going to give you an exercise to do. You can spend 5-10 minutes doing this, and it'll give me a rest and talking. I'd like you to build a model, and you can use the Slack channel or the chat to ask questions. I think a lot of people are using chat. But I want you to build a simple model of a branch system. So this is where we have one reaction that's making S1 and 2 reactions that are consuming S1. These are the rate laws, and I've got you some suggested numbers here for the constants and so on. So build that. Says run a simulation from 0 to 20 and generate 100 points and plot it. Okay? Okay, over to you. See if you can do that. 5-10 minutes. So imagine the 2 tank models. The last tank, the water just flowed out, but I didn't really care where it flowed to. It just went out in the drains out to the river, I guess. I don't know what happened to it. And I really didn't care. I could have put another tank, a 3rd tank under that, which just slowly filled up, but it wasn't what I wanted to do. So I'm doing the same thing here. Whatever's here, I really don't care. Now, if you want, you could be explicit, of course. And I could say, well, it's called waste. I'm going to call it waste because it's not something I'm really interested in, but I'll name it, right? I'll name it waste. Now, the only issue here is if I run it, of course, everything ends up in waste. And I'm really not interested in the waste. So one way around it is to just fix it at 0, right? So it never appears anywhere. So I'm back to what I was before. On the other hand, I might as well just leave it out altogether because I'm really not concerned about it. But there's nothing wrong in putting it in. There's nothing wrong in leaving it out. Okay. Yes. Well, somebody asks, does that mean there's no conservation of mass? No, there is conservation of mass. What's happening here, though, you have to think of this as an open system. If I were to stretch, let me stretch let me take a linear pathway. It might be easier to imagine. Imagine this, okay? So this is, I'm typing here on the screen if you can see it. Here's a little mini pathway. This is the kind of thing you'd see in a textbook, right? X0 goes to S1, S1 goes to S2, and then S2 goes somewhere. I mean, I'll say S3 then, okay? Now, if this is a closed system, then the total mass in the system will remain constant because it's closed. Nothing comes in, nothing leaves. And if these steps are all irreversible, and I have an initial amount of x0, over time, all the mass will end up in S3. And that's the end of the simulation. Right? Not a very interesting, not a realistic one at all. Basically, the organism is dead. So what we do is we fix x0. So now this is at fixed to some value like 5, and that means stuff is pouring in to S1 all the time, continuously, okay? Because as it pours into S1, S1 goes up, and then S1 starts to pour into S3, S2, sorry, and S2 starts to rise, and then S2 starts to pour into S3. Now in this situation, because S3 is not fixed, S3 will just climb and climb and climb and climb and climb. In fact, let's why don't we do this model? It's a nice simple one. Let's actually do this model, shall we? So that's x2, that's s2, that's s3. I think I've got everything. Let's run this. Oh, I did one. Let's see. What did I do wrong here? Oh, here. That should be S3. No, I'm making lots of mistakes here. S2, S1 to S2, S2 to S3. Hope that is okay. Now if I run this, what's the error? Line 2. What have I done wrong here?
You have a bunch of stuff off to the right.
Yeah, apart from that. Okay. Okay.
Yeah.
Right. So if you look at this, let me simulate it for less time because less time. Oh, okay. This is an interesting exercise in debugging. It's not doing what I expect. It's doing something really weird. S1 seems to be going up and up and up. Looks like it's probably going to go off to infinity. What is the problem here? Well, there's a bug in the model. Let me realign this a little bit. Unfortunately, it's not as easy as the reset. It's a lot more subtle than that. The issue is this. You have to look at the rate laws. Well, first of all, you need to look at the reactions. Are they right? X0 goes to S1, S1 goes to S2, S2 goes to S3. Okay, that seems okay. Now, the kind of error you might get with the reaction is this, where I'd put S2 goes to S2. I mean, that's a silly thing, right? That's a mistake. It should be S1 goes to S2. The mistake here, though, is in this rate law. It's saying that this reaction rate is proportional to the amount of product, which is S2. That actually should be S1. The same goes for this one. That shouldn't be S3, it should be S2. Now it should work. And it still hasn't worked. What have I done wrong now? What have I done wrong here? Let me just take that out for a sec. Oops. Something is really weird here. You may need to reset. No, it's not the reset. Simulation isn't long enough. I'll try that. Oh, that's what it was. Yeah, okay, thanks. Ryan, thanks a lot. Yeah, because I was only looking at the first little bit, it looked like a straight line. Okay. But the other bugs are genuine, right? The other bugs are genuine. So let's run this for a little bit longer then, shall we? Okay, look at this. The blue and the orange seem to be reaching a steady state, but the green is going off into hyperspace. If I run this for 200, that'll be much clearer. You see that? The green is going off and off and off. Everything is draining into S3. And everything, and because X0 is pouring stuff in all the time, and it's just getting accumulated in S3. And this is another unrealistic model. In reality, myself, right, I breathe in oxygen, breathe out carbon dioxide. There are other systems on the planet that are, on the whole, keeping carbon dioxide levels constant. So we don't see carbon dioxide going off to infinity. In order to mimic that, I need to fix S3. And now it looks a bit more realistic, okay? So it's important to fix your boundaries. In nature, boundaries are generally fixed, or at least over the short term, they're fixed, okay? And that's the way you need to build your models as well. Keep your boundaries fixed. Okay. Can we put constraints on the derivatives? No, I don't think so. Let's say,
You could do it with a piecewise equation. That's what I was thinking,
oh, right. Yeah, that's an advanced question. We'll talk about that
Yeah.
tomorrow. That's a good one. Okay, remember that then, Lucian.
Yeah.
Yeah. Yeah. All right, good. Let's move on then, shall we? Okay. So Lucian has already mentioned this, but you're saying to yourself, I want to plot other stuff. I don't want to just look at S1 and S2. I want S3. I want to plot other things. And so the simulate command has a special thing called a selection list, and it's optional, and it's just a list. And in that list, you can put the things you want to go into M or to get plotted. All right. And in this case, I've named the three reactions, J1, J2, J3, and I'd like to plot J1, J2, and J3 versus time. So in the selection list, I will list the things I want to get back. And if I run that, what did I do wrong? Oh, yeah. Rerun this cell. Sorry, make sure you run your cells. Rerun the cell, and then run that. Okay, I'm doing a reset because I want to go back to the beginning. And now you see it's plotting the rates. Look at J1. J1 is blue. It's a straight line. That's because the boundary is fixed. It's pouring in stuff at a fixed rate. Because of that, everything eventually has to match that rate. It has no choice, really. Everything has to match that. And that happens as we approach steady state. All the rates reach the same level. Okay. So you can put all sorts of stuff in here, okay? Whatever you think is convenient. We can continue this. I have no problem with that. So somebody's asking, given the time, can we possibly continue this tomorrow? That's perfectly okay with me. Is everybody okay with that? I think
Yeah, we
we'll
said we
have
would
time.
end now.
Yeah,
So
we'll have time tomorrow as well. So anyway, let's end at the selection list. I think that's a good place to pause. Yeah. Any last questions though? We'll answer any last questions and then we'll close off until tomorrow.
someone had a DM to me just about like, how do you get K values? And that kind of is the question in biology. Like, hopefully someone spent the time to measure it for you. And if not, you're gonna have to do some work, I guess.
There's two ways to get the K values. You either go to the literature and find them if anybody measured them, or what we'll do a little bit tomorrow and Wednesday, model calibration. Okay. Yeah, that's the big problem in systems biology.
And just a reminder to all, the link that you used today to enter the Zoom conference will be the same link that you use all week long. And if you have any problems, you can email Hunter. You should have my email address by now. If you don't, just send me a little message and I'll make sure that I have you.
Okay, good. Yeah, so tomorrow we'll continue with this, and then Lucian will say a little bit more about some more advanced antimony, and then we'll talk about some fun stuff on bistable systems and oscillators. Okay, see you tomorrow.
Yeah. And if anyone has like things that you're musing about, put them on the Slack. We'll get to them. We might not get to them immediately, but we'll get to them eventually.
Yeah,
or
that's
we'll
right.
address
Yeah,
them tomorrow or something. Yeah.
yeah. The chat just appears. Okay, so the
Right.
Slack days. So if you have any questions, put them into Slack. Okay, good. Yeah, thanks, guys. Yeah. The recording
links
on
should
YouTube.
have been emailed. I mean, if you didn't see them, maybe check your spam folders. But they're, yeah.
The YouTube recording should go up pretty quick, I think. That's up to Hunter, I think. Hunter, do you know when
Um,
their YouTube recordings will go up?
Yeah, it usually takes a number of hours for me to get all the recordings downloaded and then re-uploaded. We know that everything will be uploaded by Tuesday of next week, but I will try to get today's and tomorrow's online. But Wednesday through Thursday, we'll have to wait through next week.
Yeah, today's one is an important one probably because it's intro. So yeah, I'd get that one up anyway.
Yes.
Are we going to have access to the Colab notebooks? Well, they're free, so.
Yep. They're all in the course materials on the Google Drive. So
Oh,
yes.
the notebooks in the Google Drive. Oh yeah, just download them.
Yeah.
They'll be there
Download them
forever
or
anyway.
copy to your own Google Drive or whatever.
They'll be there until I'm dead, okay?
Or until Google starts charging us for Google
Or
Drive
Google
space or
starts
something.
charging us, yes. Yeah. Thanks for all your questions. A lot of great questions.
Yeah. Agreed.
Okay, I'll stop sharing if I can figure out how to.
If we have no more questions, I will go ahead and end the Zoom session.
All right, thanks, Hunter.
All right. Thanks again.
All right. Be well, everybody.
Bye.